I'll pick one word in English to describe Jair, and I couldn't find a word. So, but then I found a word. Jair, do you know what the word is? This is a quiz. No, you don't know. You don't know what the word is that describes you. The word is, I don't know how to spell it actually. Mensch. Jair's a mensch. Mensch is not English; it's Yiddish. But I think it's mensch. Jair's a mensch, and it means someone who's not just a good person, but an amazing person, like the model person, the best a human could be. That's what a mensch is. It applies to a man and a woman, even though it's a little sexist. Huh? Oh, okay. Well, would you want to tell us about something, Jair? No, but that's the one word that I found. So you failed your test, but you're still a mensch. So Jair probably thought I would talk about swimming of microorganisms because I've spent some time thinking about that topic. But I decided my birthday surprise for Jair would be not to speak about that at all, something else. And there's a reason I chose this topic, is because I knew that Jair and Stefanella had been working, and now I hear Alejandro and others, have been working on these pairs of vortices. And by coincidence, last year, I and other people, mostly other people, made a discovery about pairs of vortices in the cell, inside of living cells, inside of every one of your cells, almost every one, not the red blood cells, it turns out, but inside of all the cells of eukaryotic organisms, there seems to be something like pairs of vortices. And I'm going to describe that in the talk today. So I'm coming from Santa Barbara. Santa Barbara has an institute called the Kavli Institute for Theoretical Physics. It's sort of like IMPA in that it's beautiful and stuff like that. I invite you all to come, but don't just show up and expect I'm going to give you a place to stay. Let me know first, okay? My word, I did that already. Okay, so in the 19th century, I picked this kind of 19th century quote from Jules Verne because in the 19th century there was this dream I think scientists had, and maybe it started earlier. If we could see everything in the Earth, if we could go everywhere, every corner of the Earth that wasn't explored, and catalog and describe everything, then we'd finally be able to make theories that would describe things that seemed, that didn't have any theory. And that worked in some cases, like Darwin, for instance. It's certainly the voyage of the Beagle, that led Darwin to basically solving biology, right? He figured out where things came from. Big mystery before that, figured it out. Well, can we do that for the cell? That is, if we could catalog everything inside of cells, could we then understand how cells work? And it's amazing. Microscopists have been working at seeing everything inside of cells in different ways for decades, if not over centuries. And there's actually an organelle in all eukaryotic cells called the endoplasmic reticulum. The endoplasmic reticulum, well, it's a complicated word, but I hope by the end of my talk you'll grow to love the endoplasmic reticulum. In fact, I'm not going to say the endoplasmic reticulum many more times because everyone calls it the ER. It's a shame because endoplasmic reticulum is a great thing to say, but everyone calls it the ER. And in fact, the reason they do that is partly people don't use this term endoplasm and ectoplasm anymore. It was used at one time. Now it's outdated. Reticulum is a great word. It's Latin. It means network or net. And in fact, one thing I ask Portuguese speakers, do you know what reticulum means? And they were like, no, I don't know what it means. But there's a word in Portuguese, rede. Rede, it's directly related to the Latin root, R-E-T, for network or net. How do you say network in Yiddish? I don't know. That's a good question. I don't know. Ich verstehe nicht. I don't know that much. I have to say I'm ignorant. But anyway, the endoplasmic reticulum is an incredibly extensive organelle. It spreads out from the nuclear surface. In fact, the nuclear surface is part of the endoplasmic reticulum. It goes outside of the nucleus, it surrounds the nucleus in a funny way, becomes a network of tubes. Oops, I can't use this as a pointer, can I? Yes, probably I can. There we go. Oh, oops, there we go. This is the tubular network. This part near the nuclear surface are sheets. This is the nucleus itself, but it's not, in this particular way of imaging, you can't quite make it out too well. It's got these three different morphologies: nuclear surface, sheets, which also are not resolved, and then a network of tubes. This particular cell is a special cell that's very flat, and the biologists love to use this cell for imaging with light, because since it's so flat, you can see things because they're smushed into 2 dimensions, at least in this outer portion. But that's not typical. Usually in cells, it's all 3-dimensional. 3-dimensional network of tubes, 3-dimensional complicated configuration of sheets, and so on. Okay, so now it doesn't want to work. What happens when it doesn't work? Oh, there we go. Now it works. Oops, too well. Okay, so this is an artist's representation of the sheets, which are sometimes called the rough endoplasmic reticulum, and the tubes, which are sometimes called the smooth endoplasmic reticulum. This is a terminology based on their appearance in electron micrographs. The rough were rough looking. That's because they were seeing the ribosomes sitting on the surface of the sheets, and the ribosomes gave a texture to that surface. Ribosomes are where all the proteins are made. So the RNA is processed by the ribosomes to become the proteins. Of course, the proteins do everything in the cell, so that's a very important role for the endoplasmic reticulum. It's the site of protein synthesis. Why is there a tubular part of the ER? It's kind of a mystery. There's a lot of mysteries about the ER. The actual geometry here, this is an artist's representation. It's one of the best I've ever seen. But it's wrong in many ways, and I'll explain how it's wrong. Here's a modern image. This is, again, electron micrograph. This is a type of liver cell called a hepatocyte. What looked like tubes there, this is a cross-section. This is the nucleus in cross-section. These things, that's tubes, they're actually sheets. But you take a cross-section of a stack of sheets, and you're going to get an array of tube-like looking things. Those aren't the tubes. The tubes in this particular cell are either pushed out to the boundary of the cell, or they're not very plentiful at all in this particular type of cell. Now, all of these structures I'm talking about inside of cells or organelles, they're membrane-bound. What is a membrane? A membrane is a double layer. Every membrane is a sandwich. The bread of the sandwich are these lipid molecules. And the meat, well, I should say, the bread is the hydrophilic head of the lipid molecule, and the meat of the sandwich are the hydrophobic tails. So the hydrophobic tails in the middle, they would rather not be near water. They'd rather be closer to themselves than anything to do with water. So they're shielded from the water by this geometry. So whatever geometry keeps the meat of the sandwich inside the sandwich is a possible geometry. Of course, the molecule itself is kind of long in one direction, and it doesn't like to be at funny angles from its neighbor. So that gives rise to certain energetics of the surface. And you'll see how interesting and mathematically beautiful that is. And I wanted, yeah, please.
What about in the middle and the meat?
Yeah.
What kind of material is that? Something like a condensed matter here? Categorize that?
Absolutely they do. They call it oily tails.
What?
Oily. Oily. Not oiler. Oily. Oily, like that. Because it's like oil. In fact, these molecules are very closely related to fat molecules. In many ways, the fat molecules and the lipid molecules are synthesized in similar pathways. So these tails, just like oil You put oil in water, you know what happens, right? The oil will close in on itself and try to create the smallest surface area possible, right? It doesn't want to have that interface. Well, here there's not really an interface. The membrane itself is the other phase, you could say. This is not an interface in the traditional sense. This is a funny phase all by itself. So it's a little different than a soap film. A soap film is really two interfaces, air, interface, water, interface, air. This is a little different. This is water on the outside, and then another thing, which is like oil in the middle, but the water doesn't see that because it sees the hydrophilic parts. Anyway, evolution, biology has decided that in the cell there are going to be surfaces. And these surfaces are almost universally made out of this basic building block. That is, these molecules in a certain region of their phase diagram come together in two-dimensional sheets. And these two-dimensional sheets, of course, they bend in three-dimensional space, they connect, they have interesting geometry, interesting topology, but all of those are secondary to the fundamental structure here. And I wanted to make a point about length scales, but I didn't put it in my slides, so let me just do it now. If you remember Kurt's talk, he was talking about bacteria, so that's my picture of the edge of a bacterium. That's about 1 micron, let's say. 1 micron. Okay, so that's about 1,000 nanometers, right? So this would be 500 nanometers. This would be 250, well, let's see, 500 nanometers. 250, I'll just keep going until I hit bad numbers. Okay, 250, 120, let's say 128. Okay, 120. 60, 64, 32, and so on. I'm getting down. 16, 8. Okay, this will be my last one. 4. Okay, that little distance there, which you can hardly see, right down there, that's the thickness of one of those. That's the thickness of one of those bilayers, 4 to 5 nanometers. So, if this is the scale of the things that Kurt was speaking about, bacteria, swimming, then the scale of the thinness of this bilayer is way down here. You can't see it from the back seats. All right. So this is what I just told you. There is one extra caveat. I'll be talking about these endoplasmic reticulum ER sheets. The ER sheet is made out of the bilayers, like I said, but it's something else. It's something additional. This is the bilayer that's 4 to 5 nanometers in thickness, but it curves around and has a very high curvature. This is a cross-section. In the middle is a fluid space called a lumen. Lumen is a word that biologists use all the time. A lumen means an enclosed space, a space that you can't get to from the cytoplasm inside of cells. So that lumen plus the bilayer and another bilayer, and then this high curvature where the bilayers turn around, that's what a sheet is. So sometimes I'll talk about membranes. Sometimes I'll mean the actual two-dimensional, nearly two-dimensional bilayer. Sometimes I'll mean the sheet. The sheet, I'll reserve this word sheet to mean this sandwich, which is really a tetralayer because it's got 2 bilayers. You could say that they're connected, of course, but in the middle there's something extra too. So it's a sandwich. It's a different kind of sandwich. In this sandwich, the bread is the bilayer and the meat is the fluid in the middle, the lumen. Okay. So I'm not going to go through this whole list, but I'm going to come back to it later. These are outstanding questions about the ER. I just looked around and wrote down some of the questions that people have, the biologists and others, about the ER. Almost none of these things are answered. So I'll come back later and I'll talk about if we have any answers to those questions. But you can see there's a lot of issues about the ER. It's going to be funny for a math meeting. I thank you for inviting me here, by the way, because I'm not a mathematician, I'm a physicist. But I'm going to talk about data. I'm going to talk about how we know what we know. And I think it's important because I said there was a discovery that was made last year that I was a minor part of. The real heroes are the microscopists who did a huge amount of work. And unlike people like physicists like me, I'm standing on the shoulders of mathematicians and physicists for centuries. The microscopists, the experimentalists, have to go in the lab and from scratch make a new experiment every week or sometimes. They start from scratch sometimes, and they're amazing. So I'm going to talk about the data. So I'm going to go back to the 1930s. There was a guy in Germany named Ernst Ruska. Ernst Ruska, it's not a name that many people know, but he did win a Nobel Prize. And this is one of his images that he produced, I think, in the 1930s. While his compatriots were terrorizing their neighbors in Europe, he was doing some amazing science. Well, Jair, I'm going to ask you another quiz. What is shown in this image? What do you think? It looks just like a bunch of dirt, right? Yeah, I would just throw that one away if I was. But Ruska didn't. Well, I should say, Ruska invented the electron microscopy. He's one of the inventors. I should have said what he did. But he and his advisor, his name was Mark, I think, or Marx, invented it. Well, this is the first image I know of showing a virus. These are bacteriophages. And this is a bacterium. This is Kurt's E. coli, or this is Kurt's bacteria there. And you can see the bacteriophages that were captured here. Well, there's one of them right on the surface of the bacterium infecting it. So this is an amazing image, actually, just because the scale of the virus is really measured in nanometers. The scale of the bacterium is measured in microns, like I have over here on the board. But this showed the power, the amazing power, the ability of this electron microscopy, the new invention, to see things that have never been seen before. Images like this changed biology. Really, it came from people with physics and physical science backgrounds. This was the real revolution that changed biology in the 20th century. Another thing to notice, this is not very well resolved. Why? Because it's too big. Already a micron is too big. This thing was too big in the vertical direction, let's say, to get any resolution at all. Okay, I'm going to jump ahead 20 years. Keith Porter, American scientist who is associated with Rockefeller University mostly. He did not win a Nobel Prize. I think he should have gotten a Nobel Prize. He took these techniques he basically made the techniques in electron microscopy that are used today even to see the fine structure inside of cells. And he invented, well, this is one of his images. This shows the nucleus, the ER. Well, you can't see the ER. I can barely see it. It's a very light gray structure. And then these dark things are mitochondria, actually. These dark squiggles are the mitochondria inside of cells. But underlying that, everywhere, is a network. That's the tubular network of the endoplasmic reticulum. He was the first person to call it the endoplasmic reticulum. You see, he wrote ER there. It's this lacy structure. No one had ever seen it before. No one suspected it existed. Yet we know it's incredibly important. That's just a magnification of that. So this is a device he invented. He and Bloom. This is called the Bloom-Porter microtome. It's called a microtome because it actually takes a tissue and it slices it. Physically makes a slice. It's like those meat cutters at the delicatessen. Do you have those here in Brazil? Delicatessens. They take a sausage or something. They go shoop, shoop, shoop. This does that. This is how he made those images. This is the ancient one. This is early 50s, 1953 or something. This is the modern version. The modern version, this was pioneered by a guy at Harvard Medical School. His lab, his name is Lichtman, Jeff Lichtman. His lab has done some amazing things in refining this technique. This is where the sample sits. The blade, you can't see the blade. It's right under there. This is a conveyor belt. The conveyor belt has an adhesive tape. What happens is you slice, the adhesive tape picks up the slice. You do another slice, It goes on the adhesive tape, and so on. But that's a problem for electron microscopy. Why is that a problem for electron microscopy? Because even if this is the size of your slice, and by the way, they can slice about 27 nanometers. Let's just call it 25 nanometers in thickness. Well, what's the size of the adhesive tape? Oh, it goes off the board, right? You can't do electron microscopy on something that thick. So what they do is they don't do, there's this thing called transmission electron microscopy, where the electrons go through the sample. You can't do that. So what they do is scanning electron microscopy. They have a detector up here somewhere, and they look at the backscattered electrons from the surface. So they're resolving the surface features on this slice. Now, why is that good enough? Well, it's not good enough. You'd like to know what's inside the slice, of course. But if the slice is thin enough, 25 nanometers, you say, okay, forget it. I'm going to have another slice in 25 nanometers, so I don't care what I missed in that little thickness. I can do scanning electron microscopy on the surface, and I'll still see enough detail on the next that I can put the slices in registry and look at the three-dimensional structure of whatever I'm slicing. All right, this is just more of the setup. So I'm actually going to show you data. So I actually have to figure out how to switch out of this just for a second to show you the actual data. The actual data is so amazing, I want to show it. I'm looking at it in a program called ImageJ. Let me just see. Oops. I'm not. That's good enough. Okay, so this is one slice. And I'm going to go through there's 120-something slices. I'm going to go through them like a movie. So this is looking at a cell. This is one cell. This is inside of one cell in a brain of a rat. So this is a neuron. I'm going to do backwards now. This is a neuron inside of a rat brain. And everything you're seeing here is membrane. These are all the membrane structures. Now I should say, this at the beginning, actually, this is one cell here. And then there's other cells impinging on this one cell. You'd think that's a nucleus, but no, that's a mitochondrion in cross-section. So those early images, if you remember, Keith Porter had, showed this little dark squiggles. Those were mitochondria. This is what we can do now. This is the cross-section of one of those little squiggles. And you can see it's all filled with membrane. That's why it's so dark. This is a stain they're using that picks out only membrane. But the endoplasmic reticulum here are these squiggles. So keep your eye on these squiggles. You'll see as I step through, this is not a movie, this is just a snapshot of the cell, just frozen in time, and now I'm just slicing it. Those squiggles, at any given slice, they make equally spaced or almost equally spaced sheets. So these are slices through sheets. But the question is, how is one sheet connected to another sheet? What do these things look like in 3 dimensions? I neglected to mention that the reason they say the ER has 3 morphologies is because the biologists had discovered that it's all connected. They can release a chemical that diffuses. They could release it at the surface of the nucleus, and it will diffuse through the ER, through the complete structure, so they know it's all connected.
Just the scale of the nature, there is no distortion, there is no control.
Nope. There's no distortion in X and Y. And you might ask, what's the ratio in the Z direction? It's about 3 to 1. That is, 1 pixel here is about 3 pixels in the Z direction. So as I go from step to step, that's the only distortion there. You're missing a few pixels in the Z stage, but you asked about the absolute scale. Well, this is a sheet. So a sheet is that sandwich, which is a bilayer, lumen, bilayer. So you can see the dark surface, the dark surface of the sheet, the dark line there, that's one bilayer. Then the little light interior is the lumen, and then the other dark line is the other bilayer. Okay? That structure there, I don't know, I didn't say what the scale is, but it's tens of nanometers. Maybe that's 40 nanometers, something like that. Okay, there's a lot to talk about in the data, but the short version is that this data was such high quality that it finally answered an age-old question. How are the sheets connected to each other? And the answer, which you can kind of see from this movie, is that, let's see, I got to find a good example. I just skipped one. Well, one sheet bends toward the other sheet, and there's a topological reconnection there, and then that happens again, and it happens again, and it happens like a staircase. So if I could draw it, well, I'll just do it with my hands. Here's the two parallel sheets. This one bends toward that one. Then there's kind of a topological reconnection. Then this one comes off, and then it touches one over here. Boop. There's a topological reconnection, then this comes off, and so on. That wasn't very clear, sorry. But I have some better pictures, don't worry. Yeah, that would be great, but one has to be a little careful in using optimality arguments in biology because of evolution. Not everything is adaptive to some environment or internal pressures. So one has to be a little careful about that, but it could be, and I think I'll make some speculations at the end. Anyway, mathematical modeling of these structures. Well, I said that physicists and mathematicians could contribute to this. How can they contribute? Well, the idea of bending one of these membranes, that is bending the bilayer membrane, until about 1970, it was a real complicated mess. Maybe geometric mechanicians would not think it's so bad, but ordinary physicists like me thought it was horrible. Around 1970, that all changed. Helfrich and Kahneman published papers around the same time that simplified this tremendously. What they said is, let's look at a space of all possible shapes, and we'll define a functional on that space. We'll call that, it's called now the free energy functional. And that free energy functional will be a functional that just involves the mean curvature of the surface and the Gaussian curvature of the surface. So you plug into a surface into the functional, and you get out an energy. That's the idea of any functional, of course. But what's nice about this, it only depends on the geometry, because as Kurt told you yesterday, at these small scales, if you're in a fluid environment, every motion is overdamped, very highly overdamped. So when we're talking about energies here, we don't have to worry about inertia at all. We don't care about the history of any of these. We only care about if I took a snapshot of its shape at a given time, what's happening in that snapshot. So shape is all important in this problem. But what is the functional? Okay, I have a long story about Sophie Germain. I'm not going to tell you. Come to talk to me later. She almost got the answer exactly right. But she wasn't recognized for almost getting it right, by the way. Laplace had a lot of objections to what she did. The Helfrich-Kahneman free energy functional I mentioned. This is a prototypical form, quadratic in the mean curvature h, linear in the Gaussian curvature. Of course, I don't have to tell you about the Gauss-Bonnet theorem. Often I don't have to worry about integrating over the Gaussian curvature. Sometimes I do, often I don't. I've also put in a term here which is called the chemical potential. I'm not going to talk about that too much, but you can see it acts like a surface tension. The energetic constants here, kappa and kappa g, those are the energetic costs for bending. Well, this is called the bending modulus, and this is called the Gaussian modulus. And because this is a talk in this conference, I have to talk about Euler-Lagrange equation. So the Euler-Lagrange equation for this functional looks like this. The delta here is the Laplace-Beltrami operator. H, again, is the mean curvature. You can see it's nonlinear because I have a cubic term here. And I have a term here which is basically cubic also, because H goes like, H is mean curvature, goes like 1 over length. Gaussian curvature goes like 1 over length squared. This is basically cubic. The only linear parts here are the Laplacian and the linear term in mean curvature. You can solve this equation in certain cases. I don't know anyone who's been able to solve it generally. Tubes, tubular membranes, that is the surface of a cylinder, or nearly a cylinder, which is membrane, and fluid on the inside, that's a solution of those equations. That is not a solution of ordinary elastic equations given the same circumstances. So this is a unique feature of membranes. You can pull out long tubes. I'm not going to go into why. But the junctions between tubes, well, they've got to be basically 120 degrees. Why? Because under many conditions, if the material here and the material there and the material there all is the same stuff, and it's under the same tension, and it has the same material or constitutive properties, then you can only get force balance if you have a 120-degree angle. And you always need force balance. So at equilibrium configuration, you should expect 120-degree angle junctions. And you see that often, not always, in the tubular network of the endoplasmic reticulum. So I want to go back to this image I showed before, because the tubes are connected to the sheets, and the sheets are connected to the nucleus. And it would be interesting to know how all of that works. But there is a problem. The problem is membranes are not just lipid molecules. They're chock full of proteins. And you could say that's a problem. It is. It's a problem for mathematicians and physicists who want to model these things, because there's no great way or no universally accepted way to actually look at the model, to actually model the inclusion of proteins. This is where we need to come to some consensus. I should say there's many ways, but there's no consensus. So proteins that go through the membrane are called transmembrane. Protein that sit on the surface are called, or partly way in, are called monotopic. One-third of protein coding genes are membrane proteins. Proteins are great, however, if you're a membrane, because they allow the membranes to do things. Proteins are the way membranes do things. They change the curvature of membranes, but the proteins also respond to the changed curvature. So you have a nonlinear situation of feedback here. It's like general relativity, where the mass curves space-time, but the space-time also tells mass how to move. Well, here, proteins can curve the membranes, but the curved membrane also affects what the protein does itself. So this is a lot of opportunities, and it raises a lot of deep questions, which I won't go into. There we go. In the case of the reticulum, the endoplasmic reticulum, it's been identified a protein called reticulon. Actually, it was identified many, many decades ago, but people didn't know what its role was. So there's a hypothesis, it's more than a hypothesis now, due to Rappaport, that reticulon is kind of a W-shaped protein, and if this was the center line, or the neutral surface of the bilayer, the reticulon sits sort of like that and helps the membrane have a high curvature. How does that apply to the data I showed you? So I actually this is my only cool movie. I'm sorry, Stefanelli, your movie is much cooler than mine. But here it is again. This is what one of those junctions, that topological thing I was talking about with my hands, this is how it looks when you make it out of clay. So this was very low tech. Just take a piece of clay that represents every squiggle in that slice. And now make another piece identical to the next squiggle, and then smush it all together once you've gone up a certain z height. And you get this funny junction. Now this funny junction has some beautiful symmetries. First of all the edges here, except the one in the very center, let me show you that, this one right there, that edge is real. But all these other edges are artificial because I don't have an infinite amount of clay. Well, what if I turn this structure by 180 degrees and then rotate it a little bit? Turns out I get exactly the same structure. So that's one symmetry that this has. It's also chiral. If I had the mirror image, it would be completely different. I could not put one structure in correspondence with the other one. So it's a chiral, twisted junction. What does that make you think of? Well, it made us think of a helix. And this is some other pictures. This is actual data here, and this is that clay model again. So we thought, okay, this is the real edge. It winds through the center of that junction containing the sheets. It's locally helical. Why is it locally helical? Because every space curve is locally like a helix, right? But could it be more? Could it really be part of a helix? That is, could this be a one-dimensional boundary that winds through the entire organelle? This was sort of our dream at the time. We didn't have evidence for it. We thought it was very speculative, actually, that maybe we have a single helix that winds through the entire organelle. Wouldn't that be cool if that were the case? Well, here's actual data showing, in fact, that you can get many turns. So you're supposed to read this like go from here to there, 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 blah, blah, blah. Well, this shows how that junction is actually not a localized thing, but it's actually an extended one-dimensional defect which winds through at least this portion of the organelle. So it makes you think of a polymer. Here are the sheets of the ER, and there's some kind of polymer that's controlling where the sheets are. Like, why are they equally spaced? This was a big mystery. The biologists have no insight. Why are the ER sheets equally spaced? Well, if it's a helix has a pitch. The helix has a natural pitch. If this model is right, the pitch of the helix has to correspond to the separation between the sheets. Well, how do you model something like that? Well, one way would be to collapse. So I have to draw my sandwich again. Here's the edge of the ER. This is the edge here. This is the bilayers. So this is about 4 to 5 nanometers. This, I don't know, 40 nanometers, something like that. That's considered pretty high curvature right there. But I could do a big simplification. Now I move away from looking at membranes as manifolds. I could collapse the lumen down to 0 thickness. Then I would convert this high curvature region to a single point. Okay, it's not a manifold anymore because it ends right there, but that's okay. I'll just assign the energy I had before for a manifold. I'll just add an extra term for that boundary. And I'll say the boundary is now a space curve. After all, that was just cross-section. And I'm interested in the curvature squared integrated over the arc length of that space curve. So you can do tricks like this. And we've done this with some different levels of sophistication. But it turns out the helicoid, or something derived from a helicoid, is a solution of those membrane equations, those elastic energy equations. It's not exactly a helicoid. Everyone knows what a helicoid is. You take a straight line, doubly infinite, and you rotate it as you go down in the z direction. That generates the helicoid. It's a minimal surface. A minimal surface is a great thing to have for the Helfrich energy. The Helfrich energy, by the way, mathematicians would call it the Willmore functional. It will be minimized if you can access a surface of mean curvature equals 0. That will be a minimizer. However, you may not always be able to access that particular part of the space of surfaces. But if you can, that's great. Well, here's an example of a surface with mean curvature 0 everywhere. But I'm going to modify it slightly. First of all, let's get rid of one half of that line I rotated. I'm just going to rotate a semi-infinite line around. Okay, so that's going to have a straight axis that goes in the z direction. That'll be the boundary of that half a helicoid. But now I'm going to do something else even. I'm going to say, let's carve out, let's not start at r equals 0. Let's start at r equals some special radius. And if I start at that, then, in fact, I have something that looks like a helix. That is, its only boundary is the helix. This is like your vortex, except now this vortex is sitting in three-dimensional space. Of course, this helix is the boundary of an infinite surface. And, well, this was some heroic experimental work on a different type of cell. This is no longer a rat's neuron, but this is from a salivary cell of a mouse. A salivary cell of a mouse has very great ER structures. That's because ER, as I said, is the site of protein synthesis. And what does a cell like a salivary cell do? It secretes proteins. It secretes molecules. So its business is to produce proteins. It better have a lot of ER. So this is actual data. So this is an analysis and three-dimensional reconstruction of some of that data. And you can see, this is quite amazing. This is turn after turn of a helical structure going through these sheets. So these vortices, these helices, are actually in the cell. They're in all of your cells. Well, how can a helix connect sheets? I think I've already told you, but it would be much better if you had a pair of helices, one left-handed and right-handed. Why? Because not only would the surface have mean curvature 0 then, but if I can put them together at the right distance, I can get parallel and flat sheets that would be aligned perpendicular to that central z-axis. And so I can generate this really, when we first had this realization, when we first had this realization, we thought there would be a building block. The building block would be like one of these grand staircases that you see in, I don't know, French palaces or something. Blew up. Really? Okay. I've never been there, but this is amazing, kind of beautifully, it's an amazingly beautiful way to structure. But one thing about this structure, I should go back, it's kind of staggered. That is, if I come at one angle, I'll be coming in at level n, but then I go down the staircase, I'm at level, let's say, n plus, well, no, I did it wrong. If I come in at level n plus 3/2, I go in that direction, I'm down to level n plus 1. And then I go down another, I'm at level n plus 1/2. So even though these are at equal separation, I'm offset by half a unit on the other side of this configuration. So it's like your double vortex configurations that we heard about in the last talk, but in three-dimensional space, and now they define a surface which has this funny staggered structure of the sheets. Yeah, so those people who designed those grand European palaces, they were really smart. They anticipated this structure by hundreds of years. And this is just this is a picture of some experimental computational work, you could say, where you actually look at cross-sections. This was done by Misha Kozlov and Tom Shemesh, who did some nice numerics on this. This reminded some people of helical parking garages, which you find at airports. This is kind of the American version of the Grand European Palace, right? That is, it really has where you want to keep your most precious thing, which is your car, right? And so why not keep it in a beautiful double helical structured parking garage? I actually found this on the web. This was a patent someone had for this. Well, someone made a joke. I put it in Portuguese, but someone made a joke after our paper came out last year. They said, oh, these structures are a parking garage for ribosomes. And they showed the ribosomes all queued up, ready to get into the parking garage. Okay. Anyway, I'm almost done my talk. I know everyone is very eager for me to finish, and you can put this talk out of my misery or its misery. There's many directions that one could take. This is I say this is a discovery. I really think it is. I don't take much credit myself. All the credit goes to these amazing microscopists, but the theorists have a role to play here too. And I invite you to get involved in it. One of the things is, how are these, if the defects, these helical defects or vortices, come in pairs, how are they distributed throughout the organelle? It's not the case, it seems. We can only find in the data a few instances where you have left-handed and right-handed right next to each other. They seem to be scattered all around. But if you look at the statistics, it's very close to 50% left-handed and 50% right-handed helices. So how are they distributed in space? That's a very interesting question. How are they correlated in their positions? Could you, in fact, do a kind of scaling argument? Could you say, Well, all I can get is statistics. So I'll make a little cube of the organelle. That's L by L. And as I change L, then the total chirality, or what should I call it? The helicity. I sum the helicities, plus for left-handed, or plus for right-handed, and minus for left-handed, and I just sum it in the box. Well, it should go to 0, but how does it decay to 0? So this is a plot of this is my numerics looking at the standard deviation of the helicity as a function of the size of the box. And this is just using a de Gennes type argument. If you're a physicist, you know who de Gennes was. You probably never heard of him if you're a mathematician. But a de Gennes argument is a back-of-the-envelope calculation that uses scaling laws, blah, blah, blah. OK, this is my version of a de Gennes argument. It shows a 1 over L decrease. We don't have data yet that could show deviations from this. And deviations from this are really what is expected. The positions are probably not completely random like I'm assuming here. Another speculation I want to make. How do these things form? You have this intricate structure of helical staircases or vortices in the organelle. How did it get to be like that? We only have these snapshots of the cell at one particular time. So here's a speculation. Well, three-way junctions are very common in the tubular networks. Maybe the sheets are derived somehow from the tubular networks. Well, why is that a good suggestion? I'm not sure it is, but here's the idea. Well, the edge of a sheet is like half of a tube, right? The region of high curvature where the sheet turns around is like half a tube, half a cylinder. Could it be that you add membrane material into the network, and what happens is at the junctions, the three-way junctions, you get these fattened tubes. The tube basically splits apart. It becomes two half tubes. Well, what is it energetically favorable for that half tube to do? Is it to lie in the plane, or is it to bend out of a plane? If it bends out of the plane, it's going to become a helical half tube. A helical half tube is exactly what I need to be the edge, the junction edge, if you like, of the endoplasmic reticulum. Anyway, I go back to my initial questions that I didn't read, but now I'm going to say, what's the appropriate physical or mathematical description of these different morphologies? Well, the evidence that I want to present, just based on the success story here, is that differential geometry, geometric mechanics, let's say, that is probably the right description for these structures. It's our job to help the biologists understand them. Why are there three distinct morphologies? No idea. What makes tubes, tubes, sheets, sheets, and gives a gradient in genus? I didn't talk about the topology, but it should be clear there's a big topological difference between a network of tubes where you have a hole for every cell, high genus, and one of these funny helical punctured things. Why is there a gradient in genus? Probably because the reticulon concentration changes. When the reticulon concentration per membrane area changes, then you can get tubes becoming sheets and back and forth. How do they connect to each other? It's clear, I think, from this line of argument that a tube doesn't come into a sheet like this. A tube has to come into a sheet on its edge, like that. That will minimize these geometric energies. That doesn't answer how sheets are connected to the nucleus. That's a very good question. I don't know the exact answer to that. How does the morphology relate to distinct functions? Someone already asked this way in the back. Yes. Why do they have these shapes from the point of view of biological function? What's the teleological reasoning for why? What is the end reason for why it has that shape? We can only speculate on this now. Maybe it's related to optimal packing, as you already suggested. Maybe I can get the most ribosomes per unit volume by putting them on two-dimensional sheets that are equispaced. And the helix sets that spacing. How do these structures form and reform? That's what my last speculation was about. How do you go in time? How can you change a network of tubes into a stack of sheets? They don't seem to be related to each other. But maybe there's a beautiful geometric transformation that I'm only kind of touching upon at the very crudest way here. That is, a network of tubes may be very deeply related to a stack of sheets, even though they seem to be completely distinct and not related at all. And finally, there's a new field which, well, some of us are trying to form, called evolutionary cell biology. In this field, we view it as an accident that evolution was first discovered in looking at big organisms like you and me, and elephants and whales and birds and things. Shouldn't it have made more sense for evolution to start with the cell? How do the things inside of cells evolve? Don't look at the beaks of birds. Look at the organelles inside of cells. Why do they have the shapes they have? Forget about, why the panda has a thumb. Let's look at why the cell has an endoplasmic reticulum. And so take everything you know about evolution and apply it to single cells. That would be evolutionary cell biology. That field doesn't exist right now, it turns out. It's spoken about it? The field does not exist. I can tell you it for a fact. You can't get funding for evolutionary cell biology right now. Maybe one or two people can.
Do you have old form of said, do you have seen which cells a long time ago at the elementary level? So that you can have something to work on this.
Oh. Yeah, that would be great. I think there is some. I'm not an expert in that. Cells have been detected back to 3.7 billion years in the fossil record, but the resolution of those images is very bad. You can see there's a cell there, or there's something there. You can't see the fine structure that I'm talking about. Anyway, I take your point very well. It would be great to have data like that. Maybe that data will somehow be discovered. Anyway, I'm ending my talk on that. Happy birthday, Jair.
So I guess you will have to ask question to Greg tomorrow.
That's fine. Or you can email me.
Yes, because
there's my email address.
The bus waiting for us.